study of utilization of radioactive isotopes in the investigation of biogenetic studies. How we can use different radioactive isotopes in investigating which all intermediates or which all enzymes are responsible for the formation of the end product in the biosynthetic pathway. So in this, we are going to understand in detail introduction to biogenetic investigation, tracer techniques, radioactive tracers, how to detect and carry out the assay of radio-labeled compounds. There are different methods or techniques which are available. One of them is autoradiography. Then we are going to understand about the precursor product sequence, competitive feeding, administration of precursors, sequential analysis, use of stable isotopes, isolated organs, tissues, and cells, grafts, and mutant strains. So, introduction to this particular technique or method, that is utilization of radioactive isotopes in the biogenetic pathway. There are several advances which have taken place in understanding the biosynthetic pathways, which are dependent on various specialized techniques from wider ranges of disciplines. So I can understand how this particular biosynthetic pathway has actually been formed by using different methods or techniques. So I can take help of the usage of different mutants. I can identify different isolated enzyme systems. I can utilize radioactive tracer techniques. And more recently, by application of NMR methods to metabolites, enrich with precursors containing stable isotopes, to have all greatly contributed to these developments. So by using NMR, that is, NMR is nuclear magnetic resonance method. It is one of the highly sophisticated spectroscopic methods wherein you can identify how many carbon or how many protons are present in the given sample. So over here, I'm going to take a precursor, and that particular precursor, it is going to be labeled with the isotope. So how many protons and how many carbons are present, that can be identified with the help of NMR. Some of the important techniques are used as the tools for the elucidation of biogenetic pathway. So there are different methods which are being used and to elucidate or to describe the biogenetic pathway. The four major techniques which we are going to study in detail for biogenetic studies of primary metabolite and secondary metabolites are tracer techniques, use of isolated organs, tissues and cells, grafting methods, and use of mutant strains. We are going to understand all these techniques in detail with supporting examples. Now, what are biogenetic investigations? Biogenetic investigation, the penultimate stage. Penultimate means last but one stage. The last but one stage in the biogenetic study is the isolation of enzymes involved in the pathways under consideration and the in vitro demonstration of their properties. So if I want to understand that a particular biogenetic pathway has been elucidated or is being described, possibly there is involvement of some enzymes which are responsible for the formation of several intermediates, which has ultimately lead to formation of the end product. So if I can carry out isolation of these enzymes, which are responsible in forming different intermediates, and demonstrate their properties, I can understand which intermediates possibly are involved in the biogenetic pathway. Finally, it is becoming increasingly possible to locate and clone the gene responsible for the synthesis of particular enzyme. So these enzymes, possibly they are being formed within the plant material. These enzymes, which is what you can call it as, are getting formed because of some genes which are present, and you can locate and clone the gene. Cloning is nothing but you can have the genes of similar populations. Okay, so if there is a particular gene which is present in the plant, which is responsible for synthesis of a particular enzyme, so the same gene, I can clone it. I can produce a copy of the same gene, which will be equally responsible for the synthesis of a particular enzyme. So nowadays, advancement has taken to such an extent that I can locate and clone a gene which is responsible for synthesis of a particular enzyme. Now, the principal overall pathway associated with the secondary metabolism is largely established, and enzymatic studies are currently receiving considerable attention. So it is possible that how this particular end product is getting formed, I can actually study various enzymes which are responsible for the formation of different intermediates. So a lot of research, investigation, and advanced studies have been already being carried out in this particular area. The techniques which I'm going to discuss are used for the study of secondary metabolism, and they relate principally to the search for the intermediates involved in the particular pathway. You cannot straight away say that, okay, these are the intermediates which were present and it lead to formation of the product. You have to actually scientifically validate that which intermediates were possibly responsible for the formation of the end product. So one of the most popular method in the study of biogenetic pathway is the tracer techniques. Now, what are tracer techniques? Tracer technology is nowadays widely employed in almost all branches of sciences. This, we have understood what are stable isotopes and what are unstable isotopes. Stable isotopes, elements, they exist with identical chemical properties, but they have got different atomic weights. So these are all called as the stable isotopes. For example, say hydrogen, carbon-13, nitrogen-15, oxygen-18. Okay, these are all stable isotopes. They are not going to emit radiation or energy. Okay, they are going to remain stable. Unstable isotopes are the radioactive isotopes. Over here, the nucleus may be unstable. Say hydrogen, 14 carbon, and they decay with the emission of radiation. Now, if it is possible to detect these isotopes by suitable means, then they can be incorporated into presumed precursors of the plant constituents and used as markers in biogenetic experiments. Now, there are so many different methods which are available to detect the radioisotopes. So what we can do is, we can take presumed precursors. Presumed precursors means we are assuming that, okay, this is the precursor or this is the starting material which possibly is involved in the biosynthetic pathway. Okay, so we'll be taking the presumed precursors. We are going to incorporate radioactive isotopes into presumed precursors, and possibly they can serve as markers in biogenetic experiments. So since we are able to detect these isotopes, we can use them as what you can call it as radioactive isotopes as for tracing different intermediates which are responsible for the ultimate formation of the end product. So over here, what we are going to do is we are going to take presumed precursors, and to it we are going to incorporate radioisotopes. Why radioisotopes? Because there are so many methods which have been widely established to detect these radioisotopes. So these are the classical examples of radioactive tracers. First one is the tritium. This is the isotope of hydrogen with atomic weight of approximately 3. So how the 3 number came into being? Its nucleus, it consists of 1 proton and 2 neutrons. It has a triple mass of the nucleus of ordinary hydrogen. So tritium, that is H3 or T, it is a very widely used radioactive tracers. Similarly, radiocarbon or carbon-14, it is a radioactive isotope of carbon with the atomic nucleus containing 6 protons and 8 neutrons. So it is 6 plus 8, so 14 carbon or radiocarbon. So what are radioactive tracers? So apart from tritium, apart from carbon-14, there are other radioactive tracers also, which can be used for what you can call it as identifying different intermediates in the said biosynthetic pathway. In biological investigation, the use of radioactive carbon and radioactive hydrogen is very, very common. However, to a lesser extent and for more specific purposes, sometimes sulfur, phosphorus, alkali, and alkaline earth metals also enables the metabolism of compounds to be followed in the living organism. For example, say for studies on proteins, alkaloids, and amino acids, a labeled nitrogen atom possibly will give more specific information than the labeled carbon. So depending upon the type of the metabolite, possibly say primary metabolites, say proteins, amino acids, secondary metabolites like alkaloids, I'm able to trace a biogenetic pathway more specifically with labeled nitrogen atom in comparison to that of the labeled carbon atom.